Hi, I'm Brooke Crumling, and we're going to be discussing genetic variation through sexual reproduction. This presentation provides an overview on genetic variation through sexual reproduction. By the end of this presentation, you will be able to explain the importance of the topic, differentiate between asexual and sexual reproduction, describe the processes that occur during sexual reproduction, and list the advantages of the process. I'm going to begin with the importance of the topic. There are various reasons why it is important to understand genetic variation through sexual reproduction. The first is that sexual reproduction is how we, humans, reproduce. Understanding the science behind what occurs in fertilization, which is the process of this combination of the sperm and the egg, will allow us to understand the differences amongst our population, both big and small. Therefore, it would help us conceptualize the differentiation of people amongst the whole world and explain how babies can look different from their parents. Another factor that makes this topic so important is that genetic variation leads to natural selection. Without sexual reproduction, humans wouldn't be the same today. Genetic variation in a population is going to lead to some traits being more favorable than others. Those individuals that have the favorable traits will have greater success reproducing and surviving, ultimately passing on those favorable traits. This is crucial for the survival of a species because if dramatic environmental changes occur, the existence of that species relies on those that can live in that changing environment. For instance, if someone has only 3 offspring compared to someone with 5 offspring, the individual with 5 offspring are more likely to have at least one of their offspring survive in a drastic change in environment. So therefore, their genes will be passed on to the next generation, which is ultimately the goal of evolution. To start this topic, it is important to understand the differences between asexual and sexual reproduction. Asexual reproduction occurs from a single female parent. Although many people think of bacteria when it comes to asexual reproduction, eukaryotes, such as plants, fungi, and some animals, can also reproduce asexually. Since there is only one parent involved, the offspring are genetically identical to the parent. The main advantage of asexual reproduction is not having to depend on males, but that doesn't outweigh the benefits of sexual reproduction. Sometimes, organisms can even reproduce sexually and asexually. Let's take fungi, for example. Depending on climate regimes and accessibility of compatible sexual strands, fungi can alternate between sexual and asexual reproduction, giving them the ultimate advantage. On the other side is sexual reproduction, which involves 2 gamete cells, the sperm and egg. Every sperm and egg in the world are genetically different from one another due to the process of meiosis, which we will discuss in detail later. Therefore, there are endless possibilities of egg and sperm combinations to create genetically different organisms. Each gamete cell carries 1 set of chromosomes and are considered haploid. When fertilization occurs, the new organism will have a full set of homologous chromosomes and be diploid. This process will repeat, creating the advantage of genetically different organisms every cycle. Sexual reproduction starts with the process of meiosis as mentioned earlier, which is the making of gametes. Meiosis involves 2 meiotic cell divisions. Therefore, the cell will go through prophase, metaphase, anaphase, and telophase twice. There are 2 main separation events in meiosis. Segregation of homologous chromosome pairs occurs in the first division, and the duplicated chromosomes in each homologue are divided during the second. At the end of meiosis, there are 4 sex cells that are genetically different from one another, and it has half the number of chromosomes from the original cell. For humans, for example, the original cell would have 46 chromosomes, as shown in the picture, which is the diploid number. At the end of meiosis, the 4 cells will have their haploid number, which is 23 chromosomes. This process creates sex cells that are haploid so that when sexual reproduction occurs, the offspring will have the correct number of chromosomes of fertilization. So 23 from the sperm, 23 from the egg, coming together to get 46 chromosomes for the human species. There are various versions of genes known as alleles. For example, if you look at hair color, someone could have an allele for brown hair or for blonde hair. Every individual have at least 2 alleles for each trait, one from each parent. When the sex chromosomes combine during fertilization, the alleles from both parents combine randomly to determine the traits of the offspring and is known as the law of segregation. This is the main reason why children can look different from their parents, such as 2 brown hairs can make a blonde-haired child. If both parents carry the blonde hair gene, meaning that they are heterozygous, they're still going to have a 25% chance that their child could have blonde hair. So if you look at this picture here of the flowers, it shows there has 2 individuals that are heterozygous. Big R is going to be the dominant allele, and little r represents the recessive. So you can see that there are 1 out of 4 chance that the flower there would be white, which would be the recessive trait, even though the parents would have purple. This also helps explain how siblings can look very different from one another. One of the major steps that leads to genetic variation in sexual reproduction is homologous recombination. The chromosomes from the mother and father pair up together that are the same based on the same size and same genes. When they pair up together, the chromosomes can come in contact with one another through the formation of chiasma, as shown in the image in B. This allows 2 indistinguishable nucleotide sequences to swap genetic information, therefore mixing the genes and increasing genetic diversity. The frequency of this process occurs different amongst organisms and genders. For instance, the frequency of recombination differs dramatically between females and males. This is also consistent with other organisms such as mice. If something goes wrong during recombination, it can also lead to aneuploidy, which is chromosomal abnormality. The last thing to cover for gamete production is crossing over. This occurs during prophase of meiosis I, where the paternal and maternal homologs physically exchange homologous chromosome segments between non-sister chromatids. You may think to yourself, "What? We just talked about that in homologous recombination." It's a similar process, but different. So what happens in crossing over is that a part of the chromosome literally breaks off of it and goes and joins on the non-sister chromatids, as shown in this picture. So you can see in the picture that there are 2 different homologous chromosomes and differentiated by color. So you can literally see that one of the chromosomes were broken off and replaced on the other one and vice versa. So this one's not as much as an exchange, it is an exchange, but more of an extreme exchange by the actual breaking of the chromosomes. This process is essential for creating genetically different offsprings by producing more combinations of alleles. Crossing over is also important for repairing programmed DNA double-strand breaks and reassures correct segregation during meiosis I. There is evidence that supports crossing over as a necessary action required for correct disjunction of chromosomes during meiosis I. Eliminating this process or even reducing it could increase the likelihood of random segregation of chromosomes during meiosis I, which could lead to very harmful effects. So we've been talking about all these processes that's been happening for sexual reproduction to lead to genetic variation. So how is that going to be beneficial to us? So we mentioned this a little bit earlier, but it's going to provide organisms with an evolutionary advantage. If organisms can produce offspring that are genetically different, it's more likely that at least one of the offspring, as discussed earlier, would survive in that dramatic change in environment. So that favorable trait then allows that individual to survive the change, will then be able to pass on that trait to future generations through reproduction. External pressures influences allele frequencies in a population and leaves a mark on their genome by passing on the valuable DNA that allow them to survive and reproduce in that environment. So the cool idea of this is that you can compare the genome of an organism over times, and if you can locate specific genes that link to different traits, you can see the evolutionary changes of that organisms based on that genome because of what traits are favorable and passed on and what traits are maybe essentially eliminated. One of the other major advantages we haven't really talked about yet are mutations. So if a DNA of an individual experiences mutations, those mutations will easily get passed on to future generations if they reproduce asexually. Mutations could lead to favorable or unfavorable traits in the organism. If the mutation leads to unfavorable traits, then that organism will most likely be unsuccessful at reproducing, therefore passing on that mutation, or the mutation may not even be passed on to future generations because of making genetically different offspring in sexual reproduction through the processes that we talked about earlier, such as crossing over and DNA recombination. On the opposite end, if the mutation leads to favorable traits for reproductive success, then it would persist longer in the population. Mutations are one of the biggest causes of genetic variation in general, and the effects of sexual reproduction on top of it can lead to unpredictable outcomes. This is all the main information for the topic, and now I'm going to give you some ideas on how we're going to expand on it. As many of you know, sexual reproduction is not perfect. There are many things that can go wrong in the process and lead to unpredictable results. You just learned about essential steps such as crossing over and recombination. So what happens when those steps have errors? That can be the cause of many chromosomal disorders. One of the biggest examples of this is Down syndrome. I am providing an article that can be used to expand on this idea. As shown here, oocyte development meiosis aneuploidy. It discusses the developments of egg cells and the linkage between crossing over and recombination in Down syndrome. It states that roughly 30% of maternally caused Down syndrome patients that experience error in homologous chromosome separation had unsuccessful crossovers in the affected chromosomes. This ultimately led to the aneuploidy, which is shown here in this karyotype of an individual that has Down syndrome. There are 3 chromosomes in set 21 when there should only be 2, so this individual, this female, has an extra set of chromosomes. This article also mentions that cohesion and egg cells decreases with age in women, leading to an increase of meiotic errors. What you will do is that you will read this article and write a summary on it. The last expansion concept is on DNA recombination. We mentioned this process earlier, but didn't talk much about how humans have used this process to our advantage. This article here provided gives you the guidelines used for DNA recombination and repair studies. It dives into the techniques we can use to research DNA damage that can lead to the causes of major diseases. So if you view this image here, it gives you an overview of how we can use DNA recombination to our advantage. If we take a plasmid from bacteria and then we stretch it out, which is looking at the DNA of interest, we can treat it with a restriction enzyme. Okay? And then we are able to have better control of what happens to the specific DNA recombination process, so we can literally change the different sequences of DNA and combinations and put it back so that then we can see the effects of it. So if we can identify the specific sources, we have a better chance of dealing with them. And that is all. You will read this article also and write a summary on it. And that is it. These are the references that I have used. Which are also, I put this information in the speaker notes as well so that you know where I got all of this information from on each slide. Thank you and have a great day.